So 99% still we cannot access. Okay, so this is the tool where actually the metagenomics can ultimately give that benefit. You can at least assess the genetic material of 99% of bacteria. The story was started with the very famous, the discovery of Helicobacter pylori. Actually, two Australian scientists, Marcel and Warren, won the Nobel Prize in 2005. So before a couple of years, they were, working onto the peptic ulcer, ulceritis in stomach, okay? So they're working onto the certain bacteria, maybe bacteria is responsible for the ulceritis, peptic ulcer or not. And they, after the, started in 1983, 1982, they came to know that the one bacteria is there that is known as a Helicobacter pylori. That bacteria was reside inside our stomach. Their concentration is increased. They secrete the more amount of the acid, and due to that only the ulceritis occurs. Okay, but before that discovery, okay, in medical sector, they suggest that this is due to the some, due to the some other way. So bacteria do not having any particular role. So they just give you the certain particular acid neutralizer molecule as a drug, and the entire medication was in wrong direction in that particular time where the role of the Helicobacter pylori was not discovered. So actually, as a last part of experiment, both the scientists, take or drunk the soup of the gastric juice of the patient contains the Helicobacter pylori, and they proved that yes, there was a infection of Helicobacter pylori, and they developed the ulcer. So this was very remarkable discovery in that particular terms. The unculturable bacteria can live into the body, they live in their environment, okay, and they play their role. Okay, so for that actually they won the Nobel Prize in 2005 in chemistry and physiology. So this is the entire story, okay, what we are ultimately discussed, and this was the first, and actually the things is that in 2005, a Nobel awards was governed by Swedish government, Swedish consortium of Swedish government. So the screening committee, they just give the highest merit to this discovery. But when the name was announced, okay, all scientific community, they were shocked. Why? Because this is not a breakthrough. This is just like, very ancient, old type of discovery. Okay, at that time, they do not ultimately realize the role of, the unculturable bacteria. But though now we can realize that, this is breakthrough event because in metagenomics development, okay, this is considered as a very breakthrough event in 2005. Okay. Okay, no problem. So as far as concerned with the history of metagenomes, this is the first particular paper, the review published by Joe Handelsman in 1998, and give the terminology that is metagenomics. Okay, so this was the very, milestone paper. You should ultimately read this paper to, get some of the particular different terminology. What do you mean by metagenome, metagenomics, and many others? Yeah. So this is in 1998. It was published into these journals. Okay, and after that, many different particular works has been started. Okay. So we again come back into the culturability. So you can see over here. I'm sorry, but if I think you can see over here some of the particular figures from the different habitat. Okay, like. Soil or the farm soil, from the marine environment, from the forest soil, some desert-like soil, extreme terrestrial habitat like volcanic area, hot spring, deep sea, okay, extreme water body like hot spring, okay. So you can see the culturability is not greater than 1%, okay. So that means except forest having a culturability nearly 1 or 1.5%. That means rest of the this hidden fraction of the microorganism still we cannot access, still we cannot identify. Clear? Why? Because this is the same concept is applied like Helicobacter pylori. They are viable but cannot be culturable. Why? Because we do not having a information regarding the cultivation or requirement for the cultivation of the bacteria. Clear? So that's why when you ultimately, culture through the different particular media like LB media or the starch agar or nutrient agar, whatever the particular colonies you just observe after 24 hours there, you just assume that it is only 1%. Rest of bacteria is not generally cultured one. So culturability is very less because there are those organisms that require some special kinds of the media and which we do not having such kind of the media or the recipe to prepare the media. Clear? So that's why culturability is less. So then how we can increase, or how we can ultimately access this 99% of the hidden microbial diversity. Okay? Now the question is generated, why we should ultimately explore the such hidden microbial diversity? Because what we are ultimately taking the antibiotic, 99% antibiotic coming from this particular microbial world. Okay, many particular peptides, antimicrobial peptides, many pigment, many metabolites are there, okay, which is useful for the humankind that generally derived from the only 1% of the microbes. Still we don't have access of 99. You're just assuming that how many metabolites, new metabolites we can produce from this particular diversity. So that's why there is a need to explore for the biotechnological purpose, for the industrial purpose, for the pharmaceutical purpose, for the human benefit purpose. Okay. So that's why that we should ultimately know the composition of entire microbiome of the particular habitat. Maybe our oral cavity to the sea or water sample, hot spring, river, pond, whatever. Clear? So that means which kind of the microorganism are there, or what is exactly the role of such particular microorganism in their particular habitat. Okay, so that kind of the things we can search, okay, through the dig out by the metagenome terminology, and that what we are ultimately going to discuss. This is considered as anything but the process to study the unculturable microorganism is known as a metagenomic study. Okay, so which will start with not by culturing the organism, but which will be start by taking the unit gram of the soil from the garden or the water from the river or pond, then you just directly isolate the DNA. You are not supposed to use any streaking method or any spreading method for the cultivation. Clear? Because you have to directly isolate the total DNA. So that DNA represent from the each and every cell. Maybe they are cultivable, maybe they are not cultivable one. Okay? So if you access the DNA, you study the DNA, you can know the each and everything, no? Because DNA is considered as nothing but the progenitor molecule from where actually the RNA and workforce molecule like protein will be there. And protein is generally propel our metabolism and life is prosper. Okay? So indirectly you isolate the DNA or sometimes RNA, you can ultimately access the total microbial diversity, their role, their function, the different gene product and everything. Clear? So based on that actually you have to first go through the environmental DNA isolation. Environmental DNA, that means whatever the sample you are ultimately taking, like water sample from the lakes or the hot spring or the soil sample from the garden soil or the farm soil. Okay, so that is known as nothing but environmental DNA. Clear? So and after that extraction you just having a certain particular method through which actually you can ultimately access the number of genes, gene products, diversity of microorganism based on the genes. Clear? So we will discuss this particular part later. Okay, so there is different types of the metagenomes can be classified based on for example, environmental metagenome like soil, sediment, water of the different particular habitats. Okay, so they are known as environmental sample. That's why they are known as environmental metagenome. You can see the host associated or organismal metagenome. That means, for example, my oral cavity, so this is host associated my stomach, host associated vaginal, that is host associated. So in that sense, you can rumen of the animal, host associated. Clear? Then you can see engineered metagenome, synthetic one. Synthetic, for example, you want to ultimately analyze the microbial diversity of some reactor used by the Bali government to treat the sewage water. So this is constructed, sewage water treatment plant. Okay, so in that sense, you can ultimately having a engineered metagenome. Clear? So this is based on how actually you can going to use the different particular sample. Clear? So this is the actually entire the pipeline, how you can ultimately start with start with the metagenome pipeline. So for example, the first one you can see over here, that is the total metagenome DNA isolation from the environment, like particular sample, like soil sample from the farm, from the garden. Okay. Then you just digest with the restriction enzymes. Okay. So you will have a different size fragment of metagenome, because metagenome is very huge. Understand, no? It is very large compared to the human genome also, because it is very rich with all the particular genomes present into the different particular microbiome, particular microorganism. Clear? So you have to digest it, and after that you either go through the cloning. Okay, so cloning give you the nothing but the, understand, no? Cloning, that means you can use the different particular vector, and whatever the cut DNA are there, that is clone inside the vector. That vector become a recombinant vector. Then you can express into the different particular host cell like yeast or the E. coli, or depends on the choice. Clear? And then you can ultimately screen that each particular clone. Okay? You can ultimately screen the each particular clone. And for example, in clone number 1, you find out, yes, the amylase is produced onto the starch agar. So that means your gene cloned inside, or genes cloned inside the vector contain the amylase producing, amylase producing trait. So that means from that environmental DNA, sorry. So from that environmental DNA, you can see that the amylase producing gene is available, or maybe antibiotic producing gene is available. So that means the physical product you can ultimately extract from the cloned gene or the genes. Okay, while in case of over here in sequencing, you can find out the total particular metagenome. In total particular metagenome, how many bacteria are there, based on to the some biomarker gene like 16S rRNA, like 18S rRNA, like internal transcribed spacer of rRNA. So that can be ultimately analyzed. So that means sequence analysis give you nothing but the how many bacteria are there, which types of the bacteria are there, what is the alpha and beta and gamma diversity. So that can be calculated. So this is known as a structural metagenome or taxonomic metagenome. This is known as a functional metagenome, because it gives you the certain particular traits, okay, and physical product if you are success into the cloning. Clear? So this is the metagenome size. As far as concerned with the metagenome size, I already discussed that it is so large. Maybe it is 10 to 12 to 10 to 15 base pair, that depending upon the how much microbial diversity is present into the sample. If you are taken the sample from the arid land like desert, okay, the microbia is very less. If you are taking from the forest is very rich, okay, with the microorganisms. So that depends, okay. And it is also very how you are ultimately going to isolate the total DNA from the environmental sample, because various methods are available to use the different kits. Okay, the kits either work onto the centrifugation theory, or maybe based onto some membrane. So membrane is generally helping to the extraction of DNA. Clear? And nowadays, automatic DNA extracts are also available into the market. So we are not discussing that particular part, because this is, you all know that DNA extraction is generally possible. Okay, like water extraction kit, soil extraction kit, human stool extraction kit, everything is now available. Okay, so based on that, you can ultimately, depending upon your sample, you can use such different kits, and you will be able to ultimately find out the good DNA, good quantity of DNA. Okay. Steps into the metagenomic study, especially for both approach, like sequencing approach or the cloning approach. Okay, as far as concerned, the first one, if you emphasize onto the cloning approach, you have to require some vector, some suitable bacteria or the host cell, and after that you can ultimately success to clone the particular segment of the DNA. So you can see over here the different particular vector are generally used, like plasmid, cosmid, back, yak, pack, that depends. And that size is also how much base pair they are ultimately accommodate, cloning size. Plasmid generally used for 5 kb, not more than 5 kb. But cosmid used for 35 kb. Kb means 1000. Clear? So that depends. And if you're having those particular vector, which accommodate the very long stretch of DNA, number of clone will be reduced. If you use the plasmid, okay, plasmid accommodate only 5 kb, very less, so number of clone will be increased. So that depends on your particular choice. Clear? And then that library is generally, you know, you have to express with the certain particular detection trait, like whether this particular library, that means the collection of clone, different particular bacterial cell, okay? And you have to go through the testing of that clone, whether produce any valuable metabolites like antibiotic, like antimicrobial peptides, like biocatalyst, okay? And based on that, you can ultimately screen that. Okay, but nowadays high throughput screening is available. High throughput screening, that means at a time 96 or 384 different clones can be analyzed for the multiple trait analysis, like whether they are producing the antibiotic or like organic acid, biocatalyst, other molecules. Okay, so pigments. So based on that, you can access this library, and if you will get, yes, clone number 8 give you the antibiotic like molecule, okay, then it is good. You can preserve it, and you know, then go through the analysis, study, purification, and you can enjoy your, you know, the new particular product. I think, you know, the antibiotic turbomycin A and turbomycin B, okay, the turbomycin A and turbomycin B was discovered from the clone of the metagen. Yeah, so we have and both antibiotic now we commercialize, okay, I mean, it is for the treatment of bacterial infection. Okay, so so this is what we have discussed, entire pipeline. You having environmental sample, take the sample, go through the DNA analysis, break it, go through the cloning, either for the functional analysis or maybe structural analysis, and you will have analyze the structure of gene, trait of the gene, that means the product of that particular gene, okay, and based on that you can ultimately you know, decode the entire the microbial diversity and their role, okay, depending upon the which sample you are going to select. So this is nothing but you can see the functional analysis where, you know, your clone gene is expressed by the host cell. The product is there. That product can be ultimately identified. Maybe the chances of a novel gene, or so if there is a novel gene, the novel product is also there, like novel enzymes. You know, agar. So agar is not digested by bacteria, and that's why we use for the preparation of pet cultivation of solid media. But from this clone, now we also identify the gene which produce agarase. Agarase is the enzymes which degrade the agar. So this is possible. This is possible to, you know, identify the new enzymes. Clear? And this is sequence-based insights. So this is like okay, based on sequence, you will ultimately go through the you know, to decode that sequence, whether that gene is a functional gene or pseudo gene. If they are functional, which kind of the mRNA and protein. I mean, based on prediction, based on to the prediction software. So this is also possible. Okay, sequence-based analysis. Okay, sorry for a second. No, it is not. So meanwhile, yeah, yeah, of course, of course. Yeah. Yeah, because cloning requires a certain size. But nowadays, the manual method is not available. Directly, you can purchase the kit. For example, Cosmid is registered by Epicenter Biotechnology. Yeah, 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 yeah. So when you purchase the kit from the Epicenter Biotech company from the USA, they give you the for 10 experiment, and they're having a certain particular guideline. So depend. I mean, the once you go through the RE digestion, you have to select those particular fragment which having a 30 to 40 kb length. If it is less than 30 kb, then it is not good for the Cosmate. If it is greater than 40, it is not also good. So if you use the some plasmid kit like PBR is also available, PUC is also available, they accommodate 3 to 4 kb. So you have to again digest with the different RE to produce only 3 kb. So yeah, that you have to determine based on which vector you are going to use, which vector you are going to use. If it is yak or pack, okay, or back can accommodate 100 kb. So definitely you can use the rare cutting enzymes. Rare cutting enzyme. Rare cutting means you having two types of RE is there. One is frequent cutting and another one is rare cutting. So frequent cutting, that means those particular RE, restricted endonuclease, which cut very frequently. If you're having a long stretch of DNA, they cut by like 50 times. But rare cutting means only 2 or 3 sites is available. So they cut the long stretch of DNA only 2 times or 3 times. So you will have a long stretch of DNA. So that you have to yeah, depends on which kit, which cloning vector you are going to use. So depending on that, okay. Okay, so this is a functional base. Sorry. So functional base metagenome, where what we are discussing, this is also nothing but the that functional base. Depending upon the selection of the vector, you can ultimately choose the size of your RE digest fragment. Okay, so and based on that, actually you can ultimately use it. So and this give you the the beauty of this particular approach, that you will have some a gene product like antibiotic, like biocatalyst. Okay, because ultimately you are expressed into the certain particular bacteria or the host cell. That's why. So this many novel genes, I think maybe it is not readable, but you can see over here many novel genes and the antibiotic they already discovered from the such clone. Okay, like beta lactamase, okay, and indurubin and violacin, okay, turbomycin, many others are there. So they are discovered from the soil, you can see, and the library type, that is phosmid, cosmid, and back. So because they ultimately offer you to accommodate a very long stretch of DNA. Phosmid is also used for 30, 40, 50 kb. Cosmid is also used for 40, 45. Back is used for the 100 kb. So it is easy to manage the clone, less number of. These all are ultimately the different molecules, okay, discovered from this one. Okay, limitation of the cloning, that because it is very lengthy, very laborious, very prone to contamination. A number of the problems are there with the cloning. It is not a cup of for every person. Okay, understand, no? Because you have to have a exposure of the cloning. You have to handling with the lots of DNA, lots of clone. Understand, no? So it is not that much easy. It is also time consuming. Okay, also time consuming, because once you start with the metagenome DNA, okay, and you end with the screening, at least 5 days, successive 5 days required. Okay, so and even though it is also sometimes become a lengthy, depending upon the protocol, depending upon the selection of vector. Okay, so at least 5 days generally required. So there is n number of the challenges are there. Okay, because sometimes what will be happen, once you isolate the total DNA, dominant species DNA is frequently cloned into the bacteria. I mean, frequently clone that means the dominant species having present into the sample, they having a more amount of the DNA, while those which have a very less fraction of microorganism, okay, do not represent the high amount of DNA. So this is also sometimes problem, because they are very rare one. their concentration into the DNA is very rare. Even the concentration of the rare species into the sample is also rare, very less. So this is also one problem. So, okay, missing the rare species genome is also one challenge. Second one, that is the sequence-based metagenomes. I think most of you know that Sargasso-C project. Okay, it was one of the wonderful project, first time started by Jesse Venter. I have not forget the name of Jesse Venter. He was the scientist, and he identified Sargasso-C. Sargasso-C is just like near to Caribbean island in Pacific Ocean. Okay. So they identify 180 new genes, okay, which is never reported before. And many alga species, many bacterial species, they never reported earlier. I mean, you can just have a number of publications of Sargasso-C, authored by Jesse Venter and many others. And they sampled multiple times in different seasons also. Okay? Many examples are there, but this is well-known examples of sequence-based genomes. So they identify the novel genes, novel bacterial species, okay, or other microorganism species. So this is two particular sequence-based approach how to require the next generation sequencing. In early days, okay, the pyrosequence method was introduced in 2005 by the Roche genetics like GSFLX 454. Really, it was very wonder technology at that time, okay, but now it is not available, okay, because it is costly compared to the now new available method, okay, but the quality of sequencing was very good of GSFLX 454, okay. Nowadays, the Illumina platform is largely used, okay. Many different platforms, ISEC, MiniSec, MySec, HiSec, many platforms are available. Nanopore is also there, okay. Then even Ion Torrent is also now used, okay. So, and PEC-Bio system is also available, PEC-Bio sequencing technology, okay. So, but however, most of 70% data you can see after 2005, after 2016, that is originate from the Illumina because it is widely used, okay. So this is actually nothing but the related to the earlier slide. Many technologies, GSFLX 454, that entire technology vanished one. Now no technology is available. Though it is very good technology in the terms of nothing but the sequencing quality, but the cost per mega base pair was very high. Now it is, you can see that the cost is very less, okay, because the technology advancement and that technology advancement is helping to not only metagenomics but also in cancer genomics and many others. Clear? So a summary of this major NGS platform which is available, you can see, okay, like GSF, this is 454, okay, this is MySec and MiniSec, you can see. So the read sequence is very high. PEC-Bio is also there because PEC-Bio is just like a nanopore. The sequence read length is very high, while Illumina give you the only short fragment like 350 base pair, 450 base pair, not more than that. Okay, while nanopore give you the long stretch of DNA like 10,000 base pair. PEC-Bio also give you the 10,000 base pair. Clear? So this PEC-Bio is having their own, or nanopore they're having their own advantage because you're having a short fragment of sequence. You have to go through the assembly and then annotation. While in case of over here PEC-Bio and nanopore, the assembly becomes very less rigorous because you will have a very long stretch of DNA. Yeah? So this is actually nothing but the two side of NGS. Either you choose those which give you the short read, okay, or you can choose those sequence which give you the long stretch of DNA. Okay? But in case of PEC-Bio is suffered from some accuracy. So first generation PEC-Bio sequencing, they're having a accuracy nearly 98.5. So this is not good actually. You should having a 99.999% equivalent to 100%. Clear? So Illumina having a this kind of advantage. So I think, sir, you are not supposed to tap it. Okay, okay, no problem. Okay, sorry. So this is nothing but how the pyro sequencing or pyrotech were used before the 2012 or before the 2010. Okay, now it is totally, this is I mean just a representation. For example, this is the total metagenome isolated from the soil. Okay, so you will have the number of the different particular metagenome in your preparation. Clear? And that will be tagged with the certain particular target metagenome. That means you can ultimately use the only one gene amplification, target one. For example, 16S rRNA gene, because that 16S rRNA gene is used to determine the bacterial diversity and archaeal diversity. Similarly, you can use the 18S rRNA for the fungi for the yeast. So this is known as nothing but the targeted metagenome. If you are not target the one gene, you directly go through the entire all particular gene. For example, this is target, like you can see over here, you target this one. You target only those particular genes which are 16S rRNA. Okay? If you are not targeting the one particular gene, whole gene, okay, then what will be happen? You will having a entire the sequence of whole extracted metagenome from the soil or from the water. Clear? So this is different. So in whole metagenome, what will be happen? You will have a huge data, because whatever the ATGC are there, every and each ATGC, regardless of their origin, they are decoded. Over here, only the decoding of those segment of DNA are there, which having a specific gene like 16S rRNA. So 16S rRNA is made up of 1,500 base pair, not more than that. Clear? So only this particular 16S rRNA gene present into the millions of the bacteria which is present into the sample, only this millions of the 16S rRNA will be amplified. Okay, so your sequence file becomes very less over here in whole metagenome sequencing, where actually your file size is larger one because you're having a larger data. Clear? So you can see this is the very old our publication. Okay, this is target based of crater. Crater. This is crater lunar lake. Crater means meteorite is impacted, and due to that there is a lag. Okay, so this is the fungal. You can see that 18S rRNA. You can use it. Understand? No, because it is for the fungi. It is for the fungi. I mean. Okay, so you can use ITS or 18S. So this is targeted one. Okay, this is targeted example. In whole genome. Okay, so this is one, I just escaped this particular part because later on I will cover this particular part. This is nothing but if you are having a taxonomic data, you can convert into the phenotypic data. Clear? Okay, then, sorry. Okay, this is also good. You can see if you are having a whole metagenome of offspring, okay, you can ultimately enjoy both the particular, how many bacteria are there, okay, which are dominant one, name of everything can be possible because this is structural metagenome, okay. And another one, you can also ultimately having a function, okay, function in the terms of 1 minute, okay, function in the terms of how many bacteria, sorry. Okay, so that what is the role of the bacteria into the different particular, metabolism? How many are XYZ metabolizing organisms? So that kind of the things you can ultimately identify from the functional way. Okay, so structural give you the diversity, functional give you the diversity, along with the diversity some other functional trait of the microbiome. Clear? Okay, so this is considered as nothing but the whole metagenome sequencing. Understand? Na? Whole genome or metagenome shotgun sequencing. Clear? So that will give you the nothing but the whole genome. Okay, sometimes if your genome is, if your sequencing is deep sequencing, that means you analyze your metagenome for a longer period of time. So entire the in metagenome sequencing, you will achieve or success to achieve the whole genome of dominant species, because several times it is sequenced, okay. So what will be happen in this one, might be chances this is dominant species, for example, so whole genome of that species can also be available in your metagenome data if you use the whole metagenome approach, okay. Why it is only dominated? Because it is frequently, during the sequencing cycle, it is frequently analyzed by the sequencer. So you will have a nearly whole metagenome. So you can also extract the whole metagenome of dominant species. That is known as a binning. That process is known as a binning. Okay. So binning, that means to extract the entire genome of dominated bacteria from the metagenome data, if the metagenome data is whole shotgun metagenome data. Clear? Okay, so this is nothing but the shotgun sequencing concept. Okay, so in shotgun, the DNA is broken randomly into the numbers of the small fragments, and which are sequenced using the particular chain termination or any other particular method. Multiple overlapping reads for the target DNA are received or obtained by performing the several rounds of the fragmentation and sequencing, and computer program then use the overlapping ends to different reads to assemble them into the continuous sequence. So this is just like a assembly, okay, by overlapping. Like this is overlap, okay, then again there will be overlapping. So like this kind of the things you will have. I think in earlier lecture we go through this kind of the detail, counting generation, okay. So this is best to understand. You will have a very long DNA or metagenome. You just cut it into the small, then sequence in small way, then again assemble with the software by the finished product. Okay, so this is just like by cutting the paper, and again you have to stick the paper with the glue, and again become a paper. Clear? So this is the way how actually the sequencing and metagenomic studies generally works. And for that, actually, the multiple, the programs are available. Like this is not that much easy to directly cut. You having a different size, okay, of that genome. Then go through the multiple genome portion shared with the, different particular software, and then go through the assembly. And after overlapping, you will have a finished product. Okay, after overlapping, you will have the combined construction genome product or the genome, and that will ultimately you have to supposed to analyze. Okay, maybe sometimes it give you the whole genome of one species also. Clear? So NGS platform for shotgun sequencing. This is many software. Many methods are available until 1995 to 2005. Sanger sequence was used. After that, Pyro sequence was used. And now having Illumina, Ion Torrent, Nanopore. So I'm not discussing much, but this is evolution of different sequencing platform. Okay. Okay, so this was again our old paper. Okay, where you having a community analysis. Now understand now what do you mean by community analysis? Community means structure of bacterial community. Clear? You can also go through if it is whole. This is functional one. Now understand now what do you mean by functional? How many particular genes they are related to the glucose metabolism, how are for the fatty acid metabolism, understand? Okay, how many are stress response gene, that means understand no? What do you mean, because this is from the desert, okay? So the stress is there over, okay, so microbiome are under the stress, so you can see the 4% is stress response. So this is actually the functional. If you can find out, yes, antibiotic production is also there, okay, you can search it. Yes, there is a antibiotic producing trait is there. You can again clone that DNA and go through the cloning experiment, and you may success to find out the new trait like antibiotic producing one. Clear? So analysis tool for the metagenome. So this is the basic aspect of metagenome, how it is generally works. Okay. Just before, a demonstration, like we will have a 30, 40-minute demonstration, okay, how actually the each tools are generally used, okay. So before that, I just give you the certain particular name, introduction, and how the tools is used. So that's why it is generally given over here. So online and offline tool are available, okay. If you are online, if you are offline, so these are some of the basic terminology. I'm not just escape this particular part of an offline tool. So number of the offline tools were available, but before start with this particular pipeline, I just announced that in metagenomics analysis or bioinformatics is a very dynamic field. Today you are using the XYZ software. After 2 years, the XYZ software will not be available. Other ABC software is there. So constant you have to change your protocol, change your ability to learn the new particular software. Okay, so n number of thousands of the software are available or were available, but now it is not available. Okay, because there is a changes of the database, change some algorithm, because there is a growth of the sequence. It is daily, understand, no, it is very dynamic. Millions of the sequence daily submitted to the NCBI, EMBL, and DDPG. So you can now understand now 1 year is very long period. Okay, so every day the new software version of the software they are changed one. Okay, but however the few software are ultimately used since 2008 2009. Megan is the one of the good software, but for that you have to require the high configure computer. At least 16 GB RAM is required. It is installed if you having a minimum 8 GB of RAM in your laptop. Okay, so and when you're having a such laptop, you can't do anything other assignment you cannot do. So because it takes the much computational power. So it requires the much computational power. So Megan is very good one. Clear? For purpose of diversity analysis, comparison analysis, some statistical analysis. Clear? So I have one old study, I think we have discussed, sir, but now that study we have carried out. In 2015-16, these are the dominant software. Okay, you can see the Canberra was the first software launched in 2007. Now it is not available. Okay, Sargasso Sea project. Okay, they also use the such particular software or in 2007, but now it is not available, not functional one. Okay, then there will be introduction of the many different particular EBI metagenome. Now it is available. EBI metagenome is available because it is propelled, governed by EMBL, okay, European Bioinformatics Institute under. So it is available. Rest of all are not available. Okay, and even the recently in MGRAST was also not working. Otherwise, MGRAST is the very good database for the functional analysis. Okay, so this is the main particular problems in metagenome. So this is just rating. Okay, so this is MGRAST. Okay, I will try, but I will give you the certain if you having this is MGRAST metagenomic analysis RAST server. Okay, they having a 2 server. One is for RAST. Okay, RAST is generally used for the genome analysis, and MGRAST means metagenomic RAST server. Okay, so they're having a 2 different particular RAST and MGRAST. RAST for genome analysis, MGRAST for the metagenome analysis. Clear? And for the once you want to work in this, it is not available. Not working since last 3-4 months, but maybe it will be updated. Okay, very soon. For that, actually, you have to log in. Sign in. If you are first time, you have to sign up. After that, you sign in, and then you can upload. It is very easy. No need to tutorials. Okay, it is very self-explanatory. Just like you upload your file into the Gmail, like that way you can upload over here. Yeah. Then EBI metagenomics. Okay, so this is what we have discussed, which is governed by EMBL. We having a demo, so we will be going through that particular part. So Magnify is the so submit analysis tools inside that EBI metagenome. Okay, so whenever you search, you will have a. So this is the analysis pipeline, new analysis pipeline proposed by EBA metagenome. So we will have a hands-on session. So during that period of, during that time, I will explain you. Okay, this is one codex. Yeah, this is also beautiful, very good software. One codex. Okay, for one codex, you have to again require to log in first. You have to sign up. After that, you can log in. If you're having a FASTA Q file, you just upload it within a couple of minutes. It will be available. Okay, so this is, I mean, if you don't having any knowledge, okay, you can directly use it. Only 3 steps is there. First, you have to log in. Okay, upload the sample over here. The upload sample. You just upload the sample from our computer. Only FASTA Q file is there. Once it will be uploaded, it will be analyzed over here. So no requirement of any knowledge. It is very easy software for primary analysis of the data. Clear? And this is used for the 2 purposes. One, to find out the microbial diversity, okay, structural analysis. Second, that is used for the comparative purpose. If you're having a multiple sample, okay, multiple sample, you want to compare it, this is also possible. Okay, so if you do not having a sign in, you can just go to the, in Google you type one codex, open it, okay, and log in. And after that I will, during the demonstration session, I will give you the one data file so that you can enjoy that, how actually it is work. Okay. Okay, so this is meta-gene assist. Okay, meta-gene assist. So earlier it is also there. Meta-gene assist is used for the 2 purposes. One, that is you can predict the phenotype. For example, you just take your sample from the marine area, sea coast, okay, and you want to find out how many are halophilic bacteria, salt-loving bacteria, how many are normophilic bacteria. Okay, so this is possible with this one. Okay, so this is possible with the, and you can also find out the different traits, how many are aerobic one, how many are anaerobic one. So that kind of the traits can be possible. Okay, so this is convert your taxonomic data into phenotypic. Understand, no? Into the phenotypic. You can also find out how many are disease-causing pathogens, how many non-pathogens, okay, how many thermophiles, how many non-thermophiles, I mean mesophiles. Clear? So this kind of the data you can ultimately, so this is one. Second, it is also offer the statistical analysis. If you having a multiple data and you multiple metagenome, okay, or a single metagenome, you can ultimately analyze some statistic, not only alpha, beta diversity, but like principal component analysis, KMR analysis, okay, many other such particular analysis is generally possible. So for that actually metagenesis, I will give you the demonstration because I have a file. Because to prepare the file, it takes the time, but I will give you how actually it is work. Clear? Okay, go ahead. Okay, this is the stamp. It is offline. Okay, stamp you can directly write down the stamp plus metagenome tool in Google. You can download, you can add in your computer. Okay, but for that you have to require the SPF file. The one file is there. That file you have to require. So I will again demonstrate you how actually the stamp is generally useful. Okay, because it is also give you the lots of statistic, comparative statistic. Okay, and so that you can ultimately having a different particular statistical data. Okay, and that data is valuable for your analysis, secondary analysis of your metagenome. Clear? I'm sorry. Okay, no problem. I will actually. Just then yes, this is a very good new avenue for people like you, This name is galaxy. It is really a galaxy. Okay, in galaxy, infinity star are there. No, over here there is a millions of the different tools are there. Okay, depending upon your selection, you can search from here. The software is there. You can run the software. The running status is given over here. Okay. So, for a few particular software, they are not a stand-alone. They are not online. They are integrated into the Galaxy. Actually, earlier, without the login, you can enjoy the Galaxy. Two are there, US Galaxy and EU Galaxy. Okay, two Galaxy mirror server are there. One is usegalaxy.org, another one is usegalaxy.eu. Okay. So both now require the login. Otherwise, before few months or year, it is free. Without login, you can enjoy your analysis pipeline. Okay, but the one-time login is there, no? Like we having in our Gmail. Okay, only one-time login. Once you having a password and ID, then for lifetime you can use it, no problem. And also, if you are not okay. One more thing, without login you can use, okay, but your data will not be saved. So for login, you will get that advantage. All your data analyzed, they are stored in your account. Okay. So this is good actually. Okay, I will also give you the demonstration. And this is open source, understand, no? And if you having the ability to learn, okay, you can learn lots of things. One more thing is there, they also provide a free of cost training. Number of the module training is always going on. Okay, so it is, this is just like, the like virtual classroom for bioinformatician. Okay, so not only for the metagenome, you can having a genome analysis, transcriptomics analysis, many other particular analysis, cancer genome analysis, you can also use it. Okay, because they're having an n number of the tools. Understand, no? N number of the tools are there. So you can't predict that, thousands or more than thousands of the tools are available in this Galaxy, and they are constantly updated daily. Okay, so you can ultimately use this particular Galaxy, where I just give you the certain particular demonstration like Kraken is going to use for the structural analysis. Okay, so in my project with Dr. Virajana, we are having a metagenome, and we will also going to analyze with the Kraken 2. Now the 1 is over, 2 is there. So Kraken 2 is available in this particular site. Okay. So then after the, this is Kraken 2, okay, and after the analysis, you will have a very interactive, beautiful, Corona chart. You can play with chart, you can analyze the data in the terms of dominant species, rare species, or many other. Then KIME2, actually, KIME2 is used for the host microbiome or cancer related. They can also use for environmental sample, but it is more onto the cancer microbiome or the host microbiome. Okay, and just recently launched new particular version of KIME2. Okay, and their training and everything is also available. You can see, okay, Galaxy training is constantly available into the. And it is very easy to understand. No language is not a problem because they are ultimately very slow English language with some example. So you can do not having a barrier of like language barrier. Okay, so you can enjoy this one. Okay, you just use the Galaxy training. Okay, or http training.galaxyproject.org. So you can use this one. So. Okay, this is the whole genome extraction, removal of contaminated eukaryotic genome, and then how you can extract the whole genome from the metagenome if you having a whole shotgun metagenome sequence. Okay, I'm not discussing because this is but this is only known as a binning. Okay, this is known as a binning. Okay, why we need to analyze the different software? Why we need to go through this? Because nowadays everything is generally converted based on microbiome. Okay, our microbiome impact the efficacy of medical drugs also. I think if you having a acidity or ulcer, Someone give you the omeprazole, sometimes give you the zebeprazole, okay, and some other use the some other particular you know that drugs, okay, because in case of this is best example in acidity, different person having it treated with the different drugs because they may not work because the microbiota is different. Okay, so this is the things how actually it is help. Okay. So based on gut microbiota, uh, the medical, okay, you can also use the understanding the microbiome leads to the new food variety and more sustainable crops because indirectly food, fermented food, fermented food. So that is also based on microbiota. Okay, so cocktail of the microorganism are used, whether your tofu is, I think, fermented food, no? So this is fermented by microbial consortia. You can see over here the microbes can help us to reduce the impact onto the earth by composting our waste food and maybe even by degrading the pollution, I mean contaminants. So this is also the way, nowadays many research laboratories throughout the world, they are trying to degrade the plastic polymer. So this is because this is the huge problems, single-use plastic. I mean many, so that's why there is a need to ultimately culturing the microbiota. Okay, because culturing the microbiota, this is very beautiful picture to understand, okay, where we are, where we are going to in future. Okay, for example, this is the microbiology, only culturing the few species. If you use the 16S RNA variable region like V6, V2, or V4, you can ultimately, I mean, this is considered as our environment, okay, and we use the culturing only those sections, so hardly 1%, 2%. If you go through the 16S rRNA, okay, you can ultimately enlarge this one, okay, that is known as a target metagenome. If you again use the shotgun metagenome, the full picture or near full picture can be possible, okay, because you are extracting the everything, so it is near to full picture. So this is the just comparison. This is the traditional microbiology. This is considered as this targeted metagenome, okay, and this is the shotgun metagenome. Yeah. So culturing is generally needs because now we, having a very traditional knowledge of anti van Leeuwenhoek, then discovery of different microorganisms, then there is a Winogradsky column. I mean, this all are in microbiology. It is considered as a traditional one, okay, very conventional one. So this particular approach now, modified, like you can see, rather than the isolation, you just directly go to the metagenome. So you escape the culturing, no need of culturing, directly go to the sequencing, okay. Similarly, you will have no need of such monoculture because you can ultimately through the mass spectroscopy, you can study how many molecules is produced by bacteria, okay. So LCMS is now already going to start to identify the microorganism. Yeah, in my institute, we also send culture for the LCMS for the identification of microorganism. Now it is established. So you do not need to isolate the DNA, amplify the 16S rRNA, and then send for the sequencing. Okay, you can also identify with the mass spec. LCMS based identification is already commercialized. Okay, if you're having a liquid culture, you remove the cell from the supponent, you directly go through the LCMS, okay, because you can find out how many metabolites are there. And now MSMS is available at each particular, yes, below the molecule you can also identify. So this is possible. And Winogradsky, you can high multiplex, bacterial genome sequencing, high throughput sequencing. Winogradsky is nothing but one simulation of one ecosystem into the lab. This is known as a Winogradsky column. Okay, you take the mud, you take the sediment, soil, and pond water, fill into the one cylinder, keep it for 8 weeks. Okay, you will have a gradient of microorganisms, which is really found into the lab. Okay, because the gradient is two gradients is there used. One that is the sulfur gradient, another one oxygen gradient. Because at the top oxygen is available, in bottom of cylinder oxygen is not available. So based on that, you can having a different color. So this is considered as nothing but the one kind of simulation. So rather than this one, you can directly go through the high throughput sequencing to decode the entire the microbial diversity of hot spring or water from the pond or the river. Clear? So this new concept was there, that is known as a culturomics. Okay. When you're having a metagenomic data, you can ultimately modify the certain your methods. You can increase the culturability of your sample. That is known as a culturomics. So that culturomics is help to identify the new microbiota of the human. One paper I just read from the human gut, 450 new microbial species now identified through the culturomics. Earlier it is only few one, because our gut or entire the body contains nearly 2.5, 2.7 kg of microbiota. Okay? So this concept may be defined as an analogy with the metagenomics as approach allow extensive assessment of a microbial composition with a high throughput culturing system. Okay. So this high throughput culturing system is now going to commercialize. This is nothing but there is a different robotics setup is generally used. You have to just put the sample like water or soil. Okay. The dilution, different media composition, everything will be prepared. Okay. And also different methods are used, like rather than use of the total whole concentration of media, you can dilute the media. You can dilute the media like 0.5x, 0.1x. You can also use the microchip. I mean, it is known as the iChip. It was just, it is patented one. Patent is over in last August. Okay, so now it will be available in the market. Okay. iChip, you just, there is a small chip is there. You just write down in Google, iChip for microbiology, isolation, yes. So this is new methods where actually culturability can be increased. This is nothing, one chip, that chip having a nanopore size of Petri dish. Yeah, Petri dish, like a Petri dish, but nano size. So once you dip inside the LB, okay, and then just you put into the seashore or water, any water body, put for a few weeks. So microbes get entry over there. In one well, only one microbes can be accommodated because it is so small. So in multiple, pore, each one having a one particular microbial colony after few days. Okay, so this is very helpful to identify new microbial species. This is known as the iChip. So this is used for the you can also use the different sequencing method. MALDI-TOF is also now available for the culturomics. Okay. So this was the first concept was published, microbial culturomics paradigm shift in the human gut microbiota in 2012. Okay, but now it is already practiced after 10 years. So application of culturomics, this is in disease or abnormality. Can be diagnosed with the rare microbial species like best example of Helicobacter pylori. Okay, then microbial profiling of the normal and diseased person are different one, because diseased person are immunologically compromised and they are suppressed one. So maybe he or she is looking good when they just during the working, but might be chances entire the microbiome is very fragile. Understand? No, it is not good. So, but healthy person having a good microbiota. So this kind of the things, I think, cancer microbiota, that's why it is used, because during the cancer, the microbiota is going to change. Okay. Yeah, so autism, I think, autism is the, many patients in Australia and Southeast Asia. Autism is one disease found in baby. And I think it is just like dyslexia, just like a dyslexia, Yeah, because for dyslexia, dyslexia, that means the baby having a problem in reading and writing. Understand? No, so this kind of the diagnosis is now not, because there is no test available for this disease. Yeah, there is only psychological counseling is only the way. So microbiota is already now decoded by the autism patient as well as the dyslexia. One of my friends is in Australia. Dr. Sakuntala was work over the autism during his PhD and postdoctoral. So she also sent me the data for the analysis, and we just discussed that. So those baby having a autism, they're having a certain different pattern, and his or her sibling which reside with the same family, eating the same food of the same parent, okay, they're having some different microbiota because sibling is healthy. So I mean, this is the way how actually the microbiota or culturomics is helping. So if we transplant the good microbiota from his sibling into the autism baby, might be chances some recovery will be there. I mean, these are the some hope. These are the some hope. Okay, but this is good. Actually, now it is available. You just do you know Amazon market? Okay, where the poop of a human human poop tablet is already available. Yeah, it is already available in market. So you can use it as that poop, okay, for establish the healthy gut microbiota. If you're having a bowel disease, you know, having a constipation or having a such abnormality in your gut, you can use it. Okay, so that's why, you know, culturomics is going to use, okay, because culturomics having a value. So this is comparison, okay. I'm just escaping that particular part because this is already not in. Okay, so many applications are there. I'm not going to discuss each one because much time is already over. Okay, human health. Our entire the health is generally governed by microbiota, not by we. Okay, we don't have a, I mean, direct role in our health. Okay, whether you are eating very delicious food or you are eating any raw food or fried food or junk food, okay, but microbiota is propelling our health. Okay. So this is some of the study. I'm not just going to detail, okay, but I mean, it is very old one by 2015. Some of the cancer related publication, okay, then obesity related diarrhea, okay, many others. Cancer is now top one because cancer microbiota, okay. Number of the industrial application are there, okay. Like you can search a new biocatalyst, you can go to the patent and you can sell a lots of, you know, royalty you can have. Okay, you can also find out the new molecule like tebromycin A, terbomycin B is the new antibiotic. Okay, many other antibiotic. Yes, I just forget this one. This eye chip was used by, I mean, eye chip was produced by one professor with the student. So nearly that before 20 years, because in last August, eye chip patent was over. That means 20 year is already over. So they discovered one new antibiotic, Axiobactin, and this is only the antibiotic discovered in last 25 years. Only antibiotic discovered in last 25, Axiobactin, and that Axiobactin was discovered from this culturomics approach. Okay, so culturomics, that means you increase the culturability of the microorganism with the integration of omics data. Okay, so this is also good, no? Okay, you can have a different particular novel property of molecule, like, you understand, no? So this is only graph to present how the novel molecule, okay, like they're having a different activity, they're having a different stability, different specificity, different efficacy. So based on that, you can check it. Okay, so that will give you the yes. For example, you find out the one particular protease, and that protease is different compared to other one. So you can just go through this particular novelty, efficacy, stability, activity, and the specificity. So these are some biocatalysts screened from the metagenome library and by functional based approach. You can see these are some different soil sample and many other sample, and these are some particular different alkaline pactase, lyase, amylase, and many others, cellulase, protease, many others. Okay, biofuel microbial consortia can be developed for the biofuel production because now, you know, world is demanding the alternative source of hydrocarbon. So this is also possible, okay, because many hydrolase, for example, glycoside hydrolase is generally useful. If you're having a new such glycoside hydrolase, then definitely it will be useful into the biofuel production. The environmental remediation, it is, you know, to recover from the pollution, degrade the pollution, not only plastic, like hydrocarbon, many other compound. Okay, you can see this is the recent progress in mining of rich metagenomic resources. Okay, tebromycin A, tebromycin B. Okay, many others are the indirubin. Yeah, phasamycin A and B is also from the cosmid. Okay, so antibiotic is also, I mean, these are the not only tebromycin, but you can see arginine E is also there, fusomycin is also there, many other, idacin is also there. Okay, so these are the new antibiotic from the, yeah, this is new antibiotic. Okay. Okay, I think this is maybe only 1 or 2 PPT. Okay, so this is used in agriculture. I think no need to tell because you know that agriculture inputs require the micro. Because without the microbes, human cannot be survived. Without the microbes, plant can also not be survived. Understand, no? So a plant is health, growth, cultivation is propelled by the microbiome. Okay, and of course microbial ecology, because metagenomics is the part of microbial ecology to study the microbial ecology. Okay, future metagenomics, the n number of different devices will be there. Okay, or maybe new technology platform will be available. So the day will come where we having a different technology. That technology can be helpful. Okay, you know, now real-time metal analysis is also possible. Real-time, yeah, it is real-time. No need to atomic absorption, microscope, spectroscopy. No need. India launched one, Chandrayaan, that is satellite to the moon, where yeah, that is known as a Chandrayaan. Chandra means moon. So they use the such technology, having a laser beam. That laser is generate the heat over the surface of moon. The metals and minerals will be evaporated. That evaporated metals and minerals is real-time capture. Yeah, this was the first technology. Already now it is commercialized. So they will come where we having a laser like this one. I just put over here. Okay, then there will be burning, and whatever the material or matter will be burned over there. The ions will be generated. That ion can be captured by just like GCMS. Okay, so that is possible. So the day will come where we directly go over the, no need to carry out the sampling, bring to the lab. Okay, we just having a device like this. Okay, we just burn the water or the soil, then evaporation will be there. After that, the ion will be captured, and you can have lots of different molecules and biomolecules. So this is the day will come with within 10 days, within the 10 years. Okay. Okay, thank you very much. Okay, we now switch over with experiment. If you, question. Okay, I have a question, or otherwise later on we'll discuss the question. No problem. You can have any question. You can ask me. No problem. We will discuss it. Because earlier we have discussion, there is no textbook, reference book is available for the metagenome data tool. Okay, yeah, of course you have to learn by the different videos or clips from the different platform, like EMBL having a training schedule, Galaxy having a training schedule. You have to daily visit. If you are interested in any particular tools or the software, you can ultimately learn from the video. This is only the way, because this is very fragile or dynamic one. If I write down the entire the book, okay, when it is published and available in market, all the software is become older one. So that's why this is the main problems in bioinformatics. Okay, but bioinformatics, any analysis tool, genomics tool, chemoinformatics, informatics, or any metagenomics, genomics tool, all are having the same particular problem. So that's why the book is not available. Yeah, you can ultimately access some, good review paper, okay, good review paper, if they are recently published. Okay, so this is good for your basic theorem. Okay, otherwise you have to, learn from the different particular online platform. Okay, Galaxy is one of the good platform. And one more thing is that in Galaxy, each time there is an advertisement of jobs, fellowships, and scholarship. If you are eligible, like many master students are eligible for fellowship, many bachelor students also eligible for the fellowships, and some particular fellowship they provide online. You have to work, just like in COVID duration, we are working from the home. Okay, so you can join from the Indonesia, but your data and everything will be propelled by some other institute. So this is also possible. Okay, they also provide. I mean, not only student, it is for teacher also. Yeah, young generation teacher, those who are below 35 or 45 years, they also provide many particular scholarships and this. So this is good. I mean, this is the some advertises constantly, display into the Galaxy server, because Galaxy is daily enriched with the different software, and to use that software, to prepare the community to use that software, the person like you is required. Understand, no? Yes. So whenever you just open the Galaxy EU or Galaxy ORG, both having a displaying of certain particular, scholarships or the training, like, okay, so this is also a good opportunity. Okay, any question we will discuss, otherwise, yeah, of course, you can.
Okay. Thanks for the opportunity. I wanna, yeah.
Yesterday, I think day before yesterday, because you have to require many particular metadata file, XML file, and many other metadata file. You have to also have some, details of that particular sequence. You have to, you are not supposed to directly upload the data in raw file. You have to filter it. You have to, get out the filtering, like remove the barcode, adapters. You should having a good quality. I mean, understand, no? So directly, if you having a file from the sequencer, you cannot upload directly. Okay, so this is, and second thing is that each particular data, I mean, database like NCBI or EMBL, they having their own particular way to submit it. I mean, this is not a rocket chemistry. You can ultimately just go into the website, log in. For example, EMBL having a Webin. Webin is the interface through which actually you can upload it. Okay, Webin is the interface through which you can upload your data. But it takes time, no? Because you are always in queue. Yes, because with thousands of the people, they are daily upload the data. So it doesn't mean that once you upload, that means within 5 minutes you will have. So this is not kind of things. Understand, no? Because it is also analyzed by the software. I mean that EBI, EMBL, EBI is also analyzed software. I mean filtering, then screening. If it is having any problems, then again it is rejected. So, I mean, understand, no? Okay, yeah.
Bapak Ibu dan semua yang bergabung di online, ya kita akan ke sesi exercise karena tidak bisa ya, mungkin tidak bisa share. Nanti kita akan sampaikan lewat chat ya filenya. Terima kasih.